The overall goal of the following experiment is to observe dividing cells within a developing tissue during C. elegans embryogenesis, in order to study genes controlling cell division. This is achieved by creating or obtaining a C. elegans strain that stably expresses transgenic markers that label the cells of interest. As a second step, selectively knock down a gene of interest by feeding dsRNA to L4 larvae for 24 to 48 hours. The RNAi embryos are collected from gravid hermaphrodites and transferred to an agarose pad for live imaging. Next, using wide-field or confocal microscopy, embryos are imaged to visualize cell division phenotypes. The results will show how neuroblasts divide during C. elegans embryogenesis, and the role of genes required for their division.
This method can help reveal key questions in the field of developmental and cell biology. For example, this method could reveal genes and mechanisms that are involved in regulating mitosis during tissue development, cell-cell communication, cell migration, or cell polarity.
Use transgenic worms expressing the appropriate markers, kept on control or RNAi plates for 24 to 48 hours. Pick 4 to 6 gravid adults and transfer them to 20 to 30 microliters of M9 buffer in a depression slide. To release the embryos, use a sterile scalpel. Cut the worms on either side of the spermatheca or vulva. Next,